Hi, my name is Ben, and I'm a PhD student in the Rod Lab at the University of Toronto. And today I'll be talking to you about the evolution of female mating preferences and a test of the sensory bias model. So when looking across the many animal species, a common trend that we see is that males and females drastically differ in their phenotype. And this sexual dimorphism that we observe is caused both by natural and sexual selection. But when we're thinking about sexual selection, we can kind of further break it down into both inter and intrasexual selection. And for intersexual selection, where we're looking at the interaction between members of the opposite sex, a common thing that we focus on and study is the evolution of female mate preferences, and both the evolution of the preference itself and the evolution of these preferred traits. And so there are several different models that we use to describe this evolution. And one of these models that we can test is the sensory bias model. So in this model, selection acts on the sensory system of an organism, and this selection creates some sort of bias towards specific stimuli. Once these biases are present, females will prefer males with traits that match these pre-existing biases. And so a kind of classical example of this can be seen in these xiphophorus fishes, where in some species, males have these elongated swords on their tail fins, and females prefer males with longer swords. There are also several species where males don't have these swords. However, females prefer males if they have swords artificially attached. And so one of the great things about this model is that we can test it phylogenetically and make predictions about that, because specifically the preference is always going to evolve before the trait. And so we can look at this on a phylogeny. So we can imagine three different taxa here. And so on this red dot, we have the evolution of the preference, as well as later on the tree, we have the evolution of this trait. And so we can imagine three kind of classes of different species. We can have some species where the trait and preference are both absent. We can have another subset where the trait is absent, but the preference is there. And then finally, some species where both the trait and the preference are present. And so when we're thinking about what actually causes the evolution of the preference to initiate, a very common explanation for this that we see is that it's for some foraging preference. So this can be seen in these water mites, where males produce stimuli on the water that mimics the stimuli produced by their prey, and this will attract females to these males. This can also be seen quite well known in these Trinidadian guppies. So female guppies prefer males with long wavelength colors. So this is red, orange, and yellow. And so this study from Rod et al. 2002 looked at both male and female preferences for these long wavelength objects. So they presented them with these objects, and they found that both males and females were strongly responsive and preferred long wavelength colored objects. And so one of the possible explanations for this is that in these areas where these guppies are, these bright orange fruits tend to fall into the waters, and these fruit are carotenoid rich, and these carotenoids are important both for the production of these long wavelength colors in the males, but they also provide an immunological function, and the females require them as well as they're deposited in the eggs. So both males and females are strongly attracted to orange, and it's likely because of this foraging preference for these orange fruits. So this research was further expanded by a previous member of our lab, Michael Foisey. And so he wanted to know how widespread this preference is across poeciliids. And so he used a phylogenetic hidden Markov model. And what this model did is would predict if a species had a preference for a long wavelength color. And so he was able to predict, which you can see in the phylogeny here. And so I'll explain what the colors are. So in orange here, these are species where males of that species have the color present, and the model predicted that females should have a long wavelength color preference. Additionally, in white, these are species where the color and the preference are absent. But interestingly, there are species in black, and these are species where the coloration is absent in males. However, the model would predict that females should prefer males with these colors. Additionally, Michael's work, he also combined behavioral trials with this. So for some of these species, he was able to look at their preference in a non-mating context and compare those to the output of these models. So if a species was predicted to have a long wavelength preference, he was able to then ask in a non-mating context using these behavioral trials if there was a preference. But one issue or remaining question is if this preference exists in a mating context, because the sensory bias model specifically predicts that the preferences are going to exist both in a non-mating and a mating context. And so this is what I expanded on, is I wanted to know if a species where males naturally lack long wavelength colors, if females have a preference both in a non-mating and in a mating context for these long wavelength colors. Additionally, because I was testing preferences in these two different contexts, I was then able to ask if females individually have consistencies in their preferences between these two different contexts. And so to test this, I looked at Oryzias latipes, or the Japanese medaka. And so this is a species, as you can see from this figure, that naturally lacks any predominant coloration. And most importantly, it lacks any long wavelength coloration. Additionally, it was one of the species both tested by Foisy et al., both in the model as well as in their behavioral trials. So the model predicted that it should have a long wavelength color preference. And from the behavioral trials, both males and females had a significant response to long wavelength colored objects. Finally, behaviorally, males will court females using this circle swim behavior, and females will clearly reject the males. So there's both a clear courtship and a clear rejection behavior. And so to answer these three different questions, I used three different methods. So to look at preferences in a non-mating context, I used a multicolored object trial. To look at preferences in a mating context, I used free swim mating trials. And then finally, I compared individual preferences between the two contexts by comparing the results of the first two trials. So looking at the preferences in a non-mating context, I use these multicolored disc trials, where there are 6 different discs at the bottom of a fish tank with an association zone around them. And I scored the amount of times that a female entered that zone, the amount of times at the zone, and if a female pecked at any of these different objects. And then so for these, I used 3 long wavelength colors and then 3 non-long wavelength colors. So these are the results from the trial. So when analyzing these, one of the things is that, so in a lot of the trials, females only entered a subset of the discs. And so this led to 0 inflated data. So to analyze these, I used a hurdle model, which simply, it just fits 2 different distributions to the data. So there's a 0 inflated component, and then the rest of the data is a non-zero distribution. So for the 0 inflated component, both orange and red were significant. And so this indicates that females were significantly less likely to not enter either the orange or red disc association zone. Then when looking at the non-zero distribution, there was no significant effect of color. So these results suggest, at least from the 0 inflated component, that females do significantly prefer long wavelength colors in a non-mating context. In order to look at these preferences in a mating context, I had to artificially enhance males with long wavelength colors. And so when thinking about how to add colors, I looked at other related species. And a common pattern is that males possess color on their body. So I added these colors to these fish using injected elastomers. And so I used 6 different color treatments, and I injected males with 3 long wavelength colors, 3 long wavelength colors, 2 non-long wavelength colors, and then some males were given a sham injection. So the first thing I did is I used this multi-choice divided tank trial where females were presented with 6 different males at once. And the purpose of this trial was to determine what subset of the 6 different colors would be used in subsequent free swim trials. And I didn't use these trials as a score of female preference, just because divided tanks aren't a strong assay of female preference or female choice. So using the results of these divided tank trials, I determined what 2 colors a female would be presented with in a free swim trial. So all females were presented with 2 different males. One was a male with the color, a long wavelength color, and it was her most preferred long wavelength color from her divided tank trial, as well as her most preferred non-long wavelength color from the divided tank trial. And so during these trials, the male and female were free to associate with each other, and they scored female resisted score, which is the ratio of male displays that were immediately followed by a female resistance display, divided by the total number of male courtship displays. And previous work on medaka has shown that female resistance is significantly correlated with female latency to mate, which is a common assay of female mating preference. So from these results, females were significantly more resistant towards the males enhanced with non-long wavelength colors. And so this would suggest that, one, yes, females do have preferences for certain males with artificial stripes, and moreover, they more prefer males with red, orange, or yellow stripes added. But when looking across all of these trials, so these are some of the results, or the individual level results from the disc trials, and you can see there's a lot of within or between female variation in color response. So there's a lot of variation in female preferences. So now I wanted to ask if females were consistent individually across these two different behavioral contexts. And so to test this, each female I looked at. So I took the preference scores for her long wavelength and her non-long wavelength score. And then what I did is I compared that score to the rank of that color from that female's disc trial. So I ranked all 6 colors that she was tested with in the disc trial. And then I compared the rank of that to the preference score from the free swim trial of that same color. So these are what these results look like. So these are first the results for the long wavelength colors. And I'll explain this figure. So on the x-axis is the rank of the disc. So a rank of 1 was a more preferred color, and a rank of 6 is the least preferred. And the y-axis is the female's resistance score. So the lower the score, the less resistant a female was to that color. And so you can see there's the strong positive trend for these long wavelength colors. So females were more resistant to colors that in the free swim trials that they liked less in the disc trials. However, when we look at the non-long wavelength colors, there is no significant trend going on. So overall, at least for the long wavelength colors, we could say that yes, there is some consistency within individual females for different colors. And so overall, when we're thinking back to the sensory bias model, if these results support or refute it, so the sensory bias model, to show it, we need to first show that there's a lack of the preferred trait, which is obvious in this species. Additionally, we need to determine that the preference exists in a non-mating context, which I showed here in these disc trials. And then finally, I also need to show that the preference for that trait exists in a mating context, which I showed during the free swim trials. So overall, these results would support the presence of the sensory bias model as an explanation for long wavelength color preferences in Japanese medaka. And with that, I would like to thank all of my members of the Rod Lab, as well as funding sources, and thank all of you for listening to my talk.